Good morning. In today's presentation, we'll see about the plant fibers used as surgical dressings. So this presentation is for fourth semester B.Pharm. As per the syllabus, it is coming under the fifth unit. So when you just see a fiber, there are a number of vegetable fibers are having their importance in pharmaceutical field as the surgical dressings and also used in the manufacture of artificial fibers and hemostatic dressings. So before going into the fibers which are used in the pharmaceutical field, you should know what do you mean by a fiber. It is a tissue or a cell composed of spindle shaped or elongated structure with the pointed ends. It is otherwise known as prosenchyma. These cells are having the thick wall. Sometimes they may or may not contain lignin, and they are known as fibers. In the other words, this cell wall, they are composed of almost pure cellulose or sometimes with different degrees of lignification. That is, different amounts of lignin will be present in the form of sclerotic or sclerenchymatous fibers. On the other hand, the fibers can be called as the elongated thick-walled cells with the pointed ends. The cell wall consists of cellulose, may or may not contain lignin. And these fibers, they are used as surgical dressings in the medical field. And apart from this, they are also used as or used in the manufacture of cloth or used as the filtering medium or for the insulation purpose. And when you just see these fibers, from where and all we get or the classification of natural fibers are as follows. They are obtained from the vegetable source, animal source, and the mineral source. The examples of vegetable source may be cotton, flax, jute, and hemp. The examples of animal fibers are wool, silk, and mohair. And the mineral fiber examples are asbestos and glass wool. And apart from this, there are also regenerated fibers. That means they are the semi-synthetic fibers, we can call it as. From carbohydrates, we get regenerated cellulose, otherwise called as rayon or viscose. From proteins, usually the regenerated protein fibers are called as azlons. And they are produced from the proteins present in corn, soya bean, peanuts, and milk. And one such example is eridil from groundnut, that is nothing but your peanuts. So these are all the semi-synthetic fibers or regenerated fibers. And the examples of synthetic fibers include nylon, terylene, orlon, alginate, etc. So these are all the various classification of fibers. And next we will see about the plant, that is here the pictorial representation of your fibers used in surgical dressings. So when you say the surgical dressings, they are made up of the fibers obtained from natural source or the man-made fibers, that is nothing but your semi-synthetic and synthetic. So from the natural, you have your animal source. that is protein, wool and silk usually, and vegetable source that is from cellulose, cotton, chemical wood pulp or the other fibers like your jute, flax, hemp and all. And man-made fibers, you have the regenerated, which is from cellulose, that is nothing but your viscose rayon. And synthetic fibers, polyamides, nylon and polyesters, sterol. And next we will see about the vegetable fibers. According to your syllabus, you have 3 plant fibers you have to study. That is cotton, jute and hemp. So out of that, first we will see about the pharmacognosy of cotton. The cotton, otherwise called as your raw cotton or purified cotton or absorbent cotton. Biological source. It consists of the epidermal trichomes of the seeds of cultivated species of Gossypium, that is Gossypium herbaceum, Gossypium hirsutum, Gossypium barbedens, belonging to the family Malvaceae. I'll repeat the biological source. It consists of the epidermal trichomes of the seeds of cultivated species of Gossypium species, that is Gossypium herbaceum, Gossypium hirsutum, Gossypium barbedens, belonging to the family Malvaceae. Geographical source: United States, Egypt, and some parts of Africa and India. When you just come to the cultivation, collection, and preparation of absorbent cotton, usually the cotton is cultivated by sowing the seeds. And these seeds are sown in the rows of about 4 to 5 feet in distance. Proper fertilizers are provided timely. And these cotton plants, they grow as shrubs or small trees that bear fruits. The fruits of the cotton plant are called as capsules. And they bear the fruits after the flowering. And each capsule will have around 3 to 5 locules. And in each locule, it will have many seeds which are covered by the epidermal trichomes, or otherwise called as hairs. And the cotton capsules, which is with the seeds covered by epidermal trichomes, are called as the balls. And these balls are collected when they are ripened, and they are separated from the capsules, that is the outer covering. They are dried and subjected to a machine called as ginning press for processing. In ginning press, the epidermal trichomes or the hairs are separated from the seeds. And after the separation from the seeds, the epidermal trichomes are separated into long and short staple fibers by delinters. The short staple fibers are called as linters, and they are used in the manufacturing of absorbent cotton wool, whereas the long staple hairs are called the longer fibers, and they are used in the preparation of clothes, that is, which is used in the industrial purpose. And the seeds remain after the removal of the hairs or epidermal trichomes. They are used for the extraction of cottonseed oil and preparation of the oil cake for domestic animal feed. And the cotton which is obtained by this method is called as raw cotton. This raw cotton is full of impurities like the coloring matter, fatty material, and other vegetative parts of the plant. And this raw cotton is subjected for purification by treating with dilute soda ash solution under pressure for about 15 hours. And then during this process, the coloring matter, fatty material, and other vegetable debris are removed. And the cotton obtained by this method is bleached. Bleaching is done by treatment with sodium hypochlorite or alkaline hydrogen peroxide solution or by using dilute hydrochloric acid. And after bleaching, it is washed properly with water, dried and carded. And then it is packed by rolling into sheets between the papers. And the packed cotton is then sterilized by radiations. So that is the preparation of absorbent cotton from the cotton balls. And next we'll see about the description, that is morphologically it is white in color, which is odorless and tasteless. And shape, they are like fine filaments like that of hair, and they are soft and unicellular in nature. And the size of the absorbent cotton fibers are about 2.2 to 4.6 centimeters in length and 20 to 35 microns in diameter. And next, if you come to the chemical constituents, the raw cotton consists of around 90% of cellulose, 7 to 8% of moisture, wax, fat, and little bit of oil, around 0.5%, and cell content about 0.5%. Whereas the purified or absorbent cotton contains only cellulose and 6 to 7% of moisture. So that is about the chemical constituents of cotton. And next, the chemical test. When you just take the cotton fibers and it burns with the very little smell and fumes, and they do not produce a bead. And they may produce a small white ash because of the leaves and other things it may contain. So this is the distinction from the other the artificial or mineral fibers or the animal fibers. And the next test is the test with coaxum reagent. Coaxum reagent is nothing but ammoniacal copper oxide solution. So it is the only test distinction from the other fibers of absorbent cotton. So when you just take the cotton fibers and treat with ammoniacal copper oxide solution, the raw cotton dissolves with ballooning effect, whereas, and after dissolving it leaves the fragments of the cuticle, whereas the absorbent cotton completely dissolves with uniform swelling. The first, see in the figure, you can see the first three are the normal fibers of absorbent cotton, and the middle one is after treating with coaxum reagent, the absorbent cotton dissolves with uniform swelling. And the third figure, which is the non-absorbent cotton, which dissolves by ballooning effect, leaving the cuticles. So this is the distinction test of the absorbent cotton or the cotton from other fibers. And next test, when the dried cotton is moistened with N/50 iodine and treated with 80% sulfuric acid, a blue color is produced. This is also a distinction from the other fibers, maybe from the animal or the other plant fibers or the regenerated fibers. In cold sulfuric acid, around 80% the cotton dissolves. It is distinction from the oxidized cellulose, jute, hemp, and wool. In 60% sulfuric acid, cotton is insoluble. It is distinction from cellulose, wadding, and rayon. And in hydrochloric acid at 40 degrees centigrade, it is insoluble. It is distinction from acetate rayon and also from silk and nylon. And next, it is insoluble in 5% potassium hydroxide solution, which is distinction from oxidized cellulose, wool, and silk. In another test, it is distinction from jute, hemp, and kapok. That is when the fibers are treated with fluoroglucinol and hydrochloric acid, which will not take up the red stain because absorbent cotton is devoid of lignin. So these are all the chemical tests to identify the absorbent cotton or the cotton as raw or purified cotton. And next we'll come to the uses. It is used as a filtering medium and in the preparation of surgical dressings. This absorbent cotton absorbs blood, pus, mucus, and also it prevents the infections in the wounds. And when you just see the storage of absorbent cotton, it should be stored in a cool place protected from light and moisture. And usually they are rolled between the sheets of cotton are carded and rolled between the fibers, and long storage will make the fibers brittle and makes the absorbent into non-absorbent. So that is the pharmacognosy of absorbent cotton. And next we'll see about jute. Jute, it is otherwise called as gunny, the usually fibers or the twines and ropes, what we use it for tying, and also the bags, which are used for storing, that is the gunny bags. And apart from that, nowadays in the RTP, that is jute bags, which are fabricated for the with the different designs. The ladies, those who carry the vanity bags, which is made out of jute, and also nowadays the clothing is also made out of jute. And when you just see the synonym, it is otherwise called as gunny, and the biological source, it consists of the phloem fibers from the stems of various species of Corchorus. That is Corchorus capsularis, Corchorus volitarius, and other species of Corchorus like Corchorus cunninghami, Corchorus ginodi, all belonging to the family Tiliaceae. The geographical source mainly India, in India, West Bengal and Assam. When you just see the history of this Corchorus, it is very much interesting. About 40 to 100 species of Corchorus is available in India, which are as the flowering plants. It is the plant which is native to tropical and subtropical regions, where the fibers are used as sutures and surgical dressings. And the two Indian species, especially the Corchorus capsularis and Corchorus volitarius, they are grown in the regions of Ganges and Brahmaputra valleys. For the past many centuries, this jute, it has been the integral part of Bengali culture. In the late 19th and early 20th centuries, much of the raw jute fibers were exported to United Kingdom. In 50s and 60s, when you see this, Pakistan was the world's lead for the jute production, and these Pakistan, in those days, they were earning money out of jute. That is through the export of jute to other countries, especially from the Bangladesh. That is now it is called as Bangladesh. And this jute was called as the golden fiber in Bangladesh because it was the major portion of the foreign currency for their country. And the world's largest jute trade and processing was located in Bangladesh. So that is the small history about this jute. And when you just see the jute fibers, they are, the jute plants, they are usually tall and they are annual herbs and they grow to a height of about 2 to 4 meters. Sometimes they are unbranched, and if it is branched, it has only few side branches. And when you just see the leaves of this jute plant, they are alternate in arrangement, simple, lanceolate, 5 to 15 centimeters in long, and they have the serrate or lobed margins. And these flowers are very small, about 1.5 to 3 centimeters in diameter, and they are yellow in color. They have 5 petals, and the fruits enclose many seeds in the capsule. So already we know in the biological source it consists of the phloem fibers. So these phloem fibers are usually present in the stem branches. The unbranched stem of the jute plant was cut. The straight unbranched stem branches are cut and it is tied and put into the stagnant water for about 14 to 20 days. So during which what happens? The other vegetable debris gets decayed and they get separated from the phloem fibers which are tied into sheaves. And after that period, the fibers are taken out. They are washed well with water to remove the decayed materials, and it is carded well, bleached, and it is dried and packed and sent to the market. So that is the usual preparation of this jute fiber, and this was the olden method. So here we can see the retting process is the preparation of bast fibers. That is done by 3 methods. One is the microbial or water method, what I just told, and then next one is the steaming and the mechanical process. And these jute fibers are graded according to its colors, strength, and fiber length. And usually they are somewhat white to brownish in color, 1 to 4 meters in length. And based on that, it is separated and sent to the market. So that is the preparation and the characters of this jute fiber. And next, when you just take the fiber and observe it under the microscope by treating with chloroglucinol and hydrochloric acid, it takes up the red color because this this jute fiber contains lignin. The red color is due to the presence of lignin. And each strand will have the collection of polygonal cells, and they are surrounded by a lumen with various sizes. These strands can be separated by treating with the mixture of potassium chloride and nitric acid. And next, when you see the chemical constituents, this jute fibers primarily they contain the plant materials of cellulose and lignin. Usually they have cellulose, hemicellulose and lignin. Along with this, they also have the moisture of around 12 to 13% and wax, fat, etc. And the ash value is 1%. Chemical test, when it is treated with chloroglucinol and hydrochloric acid, it gives out the pink color due to the presence of lignin, what I explained to you earlier. And next come to the uses. It is used in the manufacture of towels, padding splints, and also used as a filtering and straining medium. And this jute is used in the preparation of coarse wax. and it has a wide range of uses. It is used to make the cloth for wrapping the bales of raw cotton, and it is used for the preparation of sacks and coarse cloth. And also they are used to weave the curtains and chair coverings and carpets. And now when you see the Hessian cloth, a very fine threads of jute can be made into an imitation to silk in the textile industry, and also it is used in the making of papers. So that is about jute. And next, hemp. This hemp consists of the pericyclic fibers obtained from the stem branches of Cannabis sativa, Cannabis indica, belonging to the family Cannabinaceae. So you know very well the flowering tops of cannabis are used as the different preparations of this cannabis are called as ganja, bhang, hashish, and all those things, which contains your tetrahydrocannabinol, which is a psychotropic substance. And the geographical source of hemp, when you see, it is mainly from uh, China, Hungary, America, Germany, Switzerland, Australia, Canada, France, and Norway. Usually the hemp is grown at an altitude from Norway to the equator. So that is about the geographical source. And when you just come to the preparation of this hemp fibers, it is same as that of jute. That is, in case of jute, it is a phloem fibers from the stem branches, whereas in case of hemp, it is a pericyclic fibers from the stem branches. So the unbranched straight stem branches are cut off, and it is allowed to put it into the stagnant water for about 14 to 20 days. During this time, where the other vegetable matter in the stem branches are getting decayed and the fibers are separated out. And after this period, the tied fibers are taken out, washed well with water to remove all the debris and other decayed materials, and the fibers are carded well, it is dried, packed, and sent to the market. That is the preparation of the hemp, same as that of jute fibers. And the chemical constituents, usually cellulose and lignin. And when you just come to the uh, uses of this hemp, it is not much used in the pharmaceutical field, and it has historical uses in the preparation of printing inks, paints, varnishes, papers, bibbles, banknotes, etc. And also it is having its importance food and textile industries, and it is used in the preparation of canvas and building materials. Because of its high strength, the fibers are ideal for the preparation of specialized paper products as tea bags and industrial filters, currency papers, and cigarette papers. So that is about the hemp. And these fibers, plant fibers, usually they are used in the manufacturing of surgical dressings. Already you might have studied about the surgical dressings in your lower semesters. And these are surgical dressings are the nothing but they are as such the fibers as a single or mixed fibers, or the woven material which is used to provide the mechanical support to the affected part or to prevent the wounds from infections. And these surgical dressings may be your gauze or bandages or the plasters, which are having their importance in the pharmaceutical and medical field. So that is about the surgical fibers and the dressings, especially the plant fibers, cotton, jute, and hemp. Okay, here we'll stop the presentation. Thank you.